So today our topic is glycolysis. Glycolysis is basically a process in which a glucose, that is a 6 carbon compound, it breaks down into two molecules of the pyruvic acid, that is known as pyruvate, that contain 3 carbon in it. So basically glycolysis is a process in which a glucose, that is a 6 carbon compound, is break down into the two molecules of pyruvic acid, that is known as pyruvate, and that pyruvic acid basically contain 3 carbons. Like one molecule contain 3 carbon, second molecule contain 3 carbon. 3 carbon plus 3 carbon will be 6 carbon compound, that will be a glucose. So in glycolysis process, that is a process in which the glucose, that is of a 6 carbon compound, will break down into the two molecules of pyruvic acid that contain the 3 carbon in it. This process is also known as EMP pathway. Why this is known as EMP pathway and what is the full form of it? EMP pathway, EMP full form is Embedded Mahroof Parnas. These three were the scientists' name, Embedded, Mahroof and Parnas. These three were the scientists who worked on this process, that is a glycolysis process. So they named this process as EMP pathway, that is Embedded Mahroof Parnas pathway. This process basically occurs in the cytoplasm of the cell, and it has two phases in it. If we talk about its phases, the glycolysis process have two phases. First phase is preparatory phase. This phase is also known as investment phase. Why it is known as investment phase? Because there is an investment in it. Which type of investment? Like it has, because here two ATP molecules are consumed in this phase, so that is why two ATP molecules are invested. So that is why the two molecules are consumed here. That is why it is known as preparatory phase or investment phase. Second phase, that is oxidative phase. Oxidative phase means here the name is showing that here oxidation will occur. What is mean by oxidation? Here oxidation occur. Oxidation means a loss of electron, or we can say a loss of hydrogen. So oxidation means the loss of electron or the loss of hydrogen. So here basically in this phase oxidation will occur. So these are two phases. We'll be discussing a cycle of that phase, that how the pyruvate is made through the glycolysis process, that how the glucose compound is break down into the molecules of pyruvic acid or the pyruvate. Now this is a complete cycle from the glycolysis process start from the breakdown of the glucose and ends at the forming of a pyruvate molecule. So we'll be discussing all these, that how pyruvate molecule is formed here. First of all, the glucose that breakdowns when it converts, first of all, that is a glucose, that is 6 carbon compound sugar. So when glucose is break down or converted, ATP is utilized here, means one ATP is consumed, two molecules of ATP is consumed here. So when it converts into that, glucose remains same. In the second step, the phosphate group or the phosphoric acid attached on the sixth carbon So here on the second step, the only the, like, what is the change here? On the second step, the phosphate, the phosphoric acid is attached on the sixth carbon of the glucose. Till here, that process is known as phosphorylation process. Like, the phosphate is attached, the phosphoric acid is attached on the sixth carbon. So here that process is known as, that step is known as phosphorylation. And here the enzyme is used here, that is hexokinase enzyme. That hexokinase enzyme is used here when the glucose is converted with the phosphoric acid. When phosphoric acid attached to the glucose on the sixth carbon, that enzyme is used here. And the cell, so till here this process is known as phosphorylation. Now the glucose, now glucose is converted into the fructose, and here same, the phosphoric acid is now attached to the sixth carbon of the fructose. Now again the fructose is again converted into the same fructose 1,6-bisphosphate. Here ATP molecule 1 ADP is consumed, 1 ATP is consumed here as well, but like there two molecules of ATP were consumed. Here there will be one ATP molecule will be consumed, and fructose will convert into the fructose 1,6-bisphosphate. Here when glucose was converted into fructose, another enzyme was used here, that was isomerase. And in the first step, the enzyme that is used was a hexokinase. Now when the fructose was converted into fructose 1,6-bisphosphate, the enzyme that was used here was phosphofructokinase. This is the enzyme that converted fructose of the phosphoric acid that was attached on the sixth carbon of the fructose, was converted into the fructose 1,6-bisphosphate. Now the fructose 1,6-bisphosphate break down into two triose, that is break down into two triose, and an enzyme is used here, that is aldolase. An enzyme is used here, in every step an enzyme is used, and another enzyme, like different enzymes are used here. So when fructose 1,6-bisphosphate is break down into two triose, an enzyme is used here, that is aldolase enzyme. Now first of all, that is dihydroxyacetone. First of all, this is formed here. Then when like through the isomerization, it is converted into the two molecules of glyceraldehyde 3-phosphate with the 3-phosphate. Like first of all, the fructose 1,6-bisphosphate was break down into two triose. First of all, which one is formed? Dihydroxyacetone. This was formed, and then through isomerization, that is converted into the two molecules of glyceraldehyde 3-phosphate. And for the isomerization, an enzyme is used here, that is isomerase. For the isomerization, then these are the two molecules of glyceraldehyde 3-phosphate, because it is now converted into the two molecules of glyceraldehyde 3-phosphate. Now there will be oxidation. As I said, the first phase was preparatory phase. We will be discussing that which one was the preparatory phase and which one was the oxidative phase. As I said, oxidation will occur in the oxidative phase, so here oxidation will occur. Oxidation occurs here in this step. So as oxidation means, as I said, that it means a loss of electron or the loss of hydrogen. So here it lost its hydrogen, and when we know when hydrogen is lost, so there would be some acceptor for receiving that hydrogen ions. So there is an acceptor, that is NAD ion. That is NAD ion that is an acceptor that will accept the glycer aldehyde 3 phosphate that will lose its hydrogen. It will receive its hydrogen and it will convert into NADH. So it will convert into NADH, and an enzyme is used here, that is dehydrogenase enzyme. This enzyme means, like, dehydrogenase means the removal of hydrogen. That enzyme helps to remove the hydrogen from these molecules. So basically oxidation occurs here, there will be a removal of hydrogen. So the NAD ion basically accept these hydrogen ions and then it convert into NADH, and then it will convert into 1,3-bis phosphoglycerate. Then what will happen? 1,3-bis phosphoglycerate will convert into 3-phosphoglycerate by an enzyme, that is phosphotransferase. That is an enzyme that will help 1,3-bis phosphoglycerate to convert into 3-phosphoglycerate. Here ATP will also consumed, like 2 molecules of ATP will be formed here as well. Now the 3-phosphoglycerate is formed here. Now when 3-phosphoglycerate will convert into the simple into the 2 molecules of phosphoglycerate. Here it was on the, like, 3rd phosphoglycerate, it was 3-phosphoglycerate, then it will convert into the 2-phosphoglycerate, and enzyme will use here, that will mutase. Then the 2-phosphoglycerate will convert into phosphoenol pyrate, and the enzyme will use here, that is enolase enzyme. It will use here, and it will phosphoglycerate will convert into phosphoenol pyrate. The last step, that phosphoenol pyrate will convert into pyrate, and here water is released as well, and here ADP will convert into ATP, means 2 molecules of ATP is formed here as well. In this last step, that phosphoenol pyrate will convert into the simple pyrate. So that was all the process. Now the glycolysis will convert, as I said in the first definition, that glycolysis is a process where the glucose will break down into the 2 molecules of pyrate, means pyruvic acid, 3 carbon in both the molecules. So that is the whole process through which the glucose is converted into the pyrate. That is a cycle of the glycolysis process. In each step the enzyme is used here, and in some steps ATP molecules are also formed. Now as I said the phase, that is a preparatory phase and oxidative phase. Now which one is preparatory phase here and which one is oxidative phase, let's discuss here. Till here that is a preparatory phase. This is a preparatory phase, and 2 molecules of it, as I said, it is also known as investment phase. Why? Because 2 molecules of ATP is consumed here as well. So it is preparatory phase is this. Now after this is here oxidation will occur, so that is a oxidative phase. So there are 2 phases in the glycolysis process through which it completes.